Hi, so I'm here to talk about exometabolites in the phycosphere. So the phycosphere just means the region around algal cells. So why do we care about algae? Well, algae are really important. They account for more than 50% of global photosynthesis. They're also important industrially for bioproduct and biofuel production. But algae are never alone. So whether they're in the oceans or in these algal ponds, they're always surrounded by a complex microbial community. And that microbial community is important for supporting the health and productivity of the algae. So we want to understand what's going on in these communities, what are the interactions so that we can understand what's going on in terms of biogeochemical cycling and also in these industrial algal processes. So in these systems, we know that the algae is the primary producer. It's taking in carbon dioxide, doing photosynthesis, and producing a complex mix of exometabolites. And those exometabolites are feeding and shaping the surrounding microbial community. So we wanted to ask the question, can we use exometabolite consumption profiles for these microbes to understand the interactions in these communities? So for instance, we can have microbes that are wanting to consume the same exometabolites from the algae, and that sets up a competitive interaction. Or we could have other microbes that have different sets of metabolites that they want to consume, setting up more of a resource partitioning. And so the framework that I'm going to be talking about this in is in terms of the influence of one bacteria on another bacteria. So again, the algae is producing all these exometabolites. Our first bacteria, which I'm calling the influencer, is taking up some of those exometabolites and changing the suite of metabolites that are then available to our recipient bacteria, and how is that affecting the growth of that recipient bacteria. So the first step of this is, of course, to understand what are the different bacteria consuming from the algal exometabolites. So for this experiment, we have our algal exometabolites in the form of spent medium from our algae. We feed it to a suite of 10 different bacterial isolates. These are isolates that were derived from algal-associated community. So they are adapted to live together with this particular algae. And then we incubate those, and we did an untargeted exometabolomic analysis to look at the metabolite consumption patterns of our different bacteria. So that's what we're looking at here. So out of the hundreds of exometabolite features that we saw, these are the ones that were significantly consumed, significantly depleted by our different microbes. So on the x-axis, we have our different metabolite features. On the y-axis, we have our different microbes. And the darker color indicates more consumption, so more drawdown of that particular metabolite by a particular microbe. And so what you can see here is that the different microbes have different consumption patterns. And we can start to look at the overlap between those consumption patterns to get an idea of what interactions we might expect. So for example, if we look at this Algorophagus and Alcanophorax, they're consuming pretty much the same set of exometabolites. So we would expect this to set up competition between these different bacteria. They're trying to consume the same resources. Whereas if we look at a different set of bacteria, if we look at this Devosia and Rosibium, well, there's some overlap. We see that for the most part, they're consuming different sets of metabolites. So we would expect this to be more of a resource partitioning type of interaction. So we can actually use this overlap to quantify an expected level of competition between our different bacteria. So that's what we're doing here. We're looking at, and again, we're looking at this in terms of the influence of one bacteria consuming some metabolites on the growth of on the downstream bacteria, and what we would expect that competition to look like based on that metabolite consumption overlap. And so that's what we're looking at here. On the x-axis, we have that first influencer bacterial strain. On the y-axis, we have the recipient strain. And then the darker color indicates more competition. So dark blue means that they're competing for the same resources. The lighter blue to white means that they're sharing the resources better and each consuming different ones. And then above each column, the box plot just is a summary of the data in that column of the heat map. And so what we can see is that, again, we have different patterns for different bacteria. So for some of these bacteria, like the Alcanovorax, the Elgoraphagus, and the Sulfitobacter, they have overall pretty low, pretty negative expected competitive interactions. So we would expect them to compete for those resources and have negative effects on the other bacteria. Whereas if we look at these bacteria, the Marinobacter and the Devosia, they don't have as much overlap with the other bacteria. And so they have much less negative expected competition with other bacteria. So this is great. This gives us an expectation for what interactions we would see with the different bacteria. The next experiment, of course, was to test that. So again, we have our algal spent media. That's our exometabolites that we feed to our different bacterial isolates. In our first incubation, that's our influencer stage. So those bacteria are taking up some of those metabolites, changing the what's available. We take that spent medium and actually feed it to all of the other bacteria and see the effect on growth. And so we did this for all 100 pairwise combinations of bacteria and looked at the effects on growth. And so this is what I'm showing you here. This is analogous to the previous plot with the influencer strain and the recipient strain. But what we're looking at is the measured sequential interaction rather than the expected interaction. And so probably the first thing you'll notice here is that there's a bunch of red as well as blue. So as I said, the blue meant competition. The white means more resource partitioning and sharing of metabolites. And the red actually indicates a cooperative or a facilitative interaction, which means that that influencer bacteria is actually increasing the growth of the downstream recipient bacteria. So this was kind of exciting to see. And again, we see different patterns with different bacteria. So this Alcanovorax, again, has generally very negative impacts on the downstream bacteria, as opposed to this Devosia, which you can see there's a lot of red there, indicating that it's having generally positive effects on many of the other bacteria. And so we can compare our expected competition to our actual measured sequential interaction. So that's what we're doing in this plot here. On the x-axis, we have the expected competition, and on the y-axis, we have the measured sequential interaction. And then each of these points is a bacterial pair, so a pair of an influencer and recipient bacteria, and what we expected versus what we actually measured. And this gray line here toward the bottom, this diagonal line, is the one-to-one line where what we measured matched what we expected based on the metabolite overlap. And so what you can see is that sort of gives us a minimum. It's the worst case, most negative interaction that we see is about what we would expect from competition. And some of our points fall on that line. We don't really see anything worse than that, but we do see some that fall above that line where the interaction is not as negative as we would expect, and some of them are even quite positive. So from this, we decided that we wanted to really dig into a few of these interactions. So we're looking at Alcanovorax, Devosia, and Marinobacter. You'll notice those are some of the names that I mentioned before. And we're looking at the influence on Marinobacter growth. So as you can see, these 3 points that are highlighted here are the ones that we're testing. As you can see, this one that falls toward the bottom, that's the Alcanovorax. That's the one that we expect that we saw had a pretty negative impact, and it's falling pretty close to that expected competition line. Whereas if we look at the one near the top, that's the effective Devosia. That's the one that had very positive impacts. And so we wanted to dig further into these interactions. And for that, we turned to our collaborators at MIT, Hugh and Cullen. And they've developed this really interesting porous microplate system. And so what this is, it's a microplate. We have wells that we can grow our different organisms in, but it's made of a porous material that allows for diffusion of metabolites and nutrients between the wells. And so the bacteria are isolated from each other, but there's this metabolite exchange going on. And so we set this up with the algae in a large central well. And then adjacent to that, we have a ring of wells that has our influencer bacteria. And then outside of that, we have another ring of wells that has our recipient bacteria. And so the algal exometabolites, the algae, are doing photosynthesis. They're putting out these metabolites, and those are diffusing out past the influencer bacteria first, and then to the recipient bacteria. It's important to note that you will also have diffusion of media and bacterial exometabolites in the other direction. So we set this up with our different influencer bacteria on that inner ring, and then the outer ring with Marinobacter. And we looked at both the impact on growth, looking at cell counts, but also at single cell carbon incorporation with the nanosims. And so what we're doing here, we're feeding, we're providing 13C labeled bicarbonate. That's getting taken up by the algae. They're doing photosynthesis, and they're putting out these labeled exometabolites. And that's what's diffusing out to our bacteria. Bacteria take up that label, and when they incorporate it into their biomass, then the bacteria become labeled. So at the end of our experiment, we harvest those bacteria, and we look at them under the nanosims, and we get these lovely pictures that can tell us about the 13C enrichment of individual bacterial cells. And so for each of our conditions, we get a distribution of carbon-13 incorporation by our bacterial cells. We can combine that with our with our cell counts and get an overall picture of the 13C incorporation. And so that's what we're looking at here. And what we can see is that for Davosii and Marinobacter, so the parent bacteria that we expected to be cooperating, we see a much higher level of 13C incorporation, as opposed to the Alcanovorax and Marinobacter system, which we expected to be more competitive, where we see a much lower level of overall carbon-13 incorporation. And so what that tells us overall from these sets of experiments so far is that first, the exometabolite consumption patterns were able to give us a prediction of the competitive interactions, of the most negative interactions, but we did see these other more cooperative interactions based on our activity-based and growth-based studies. And finally, what we saw with the microplate experiment was that the cooperative system was the one that had the most algal carbon drawdown, as opposed to the more competitive system, which had a very low total carbon drawdown. And so with that, I'd like to say thank you to a whole bunch of people involved in this project at Lawrence Livermore Lab, especially Xavier, who did the nanosims work, and Courtney, who ran hundreds of flow cytometry samples for us. Of course, people here at the JGI, especially Amber, who ran all of our metabolite samples on the LCMS, and then at MIT, especially Hugh, who designed and did the microplate experiments. And with that, I'll take questions.
All right, thank you, Vanessa. So we already have a question.
Great talk. That's a lot of data that you've got. So I was wondering about growth type, or kind of growth stationary phase, and when you're adding the exometabolites. So did you standardize for that, and did you look within a strain when was best to kind of add the exometabolites into the culture?
So you're asking about the growth of phase of the algae when we can collect those?
Both the algae and the bacteria, yeah.
So both the algae and the bacteria. So in terms of the algae, we worked with this particular algae, Phaeodactyla molot, and done time course profiles of the exometabolites. And so we have it dialed in pretty well that we're collecting it toward the end of the exponential growth phase. to get the most exometabolites without seeing a whole lot of cell death. That's how we determined where to collect that. In terms of the growth of the bacteria, so we're actually, we're not adding it into an already growing culture. This is the only carbon source that we're working with. So we're adding fresh cells into this exometabolites and then waiting for them to grow in that.
Okay, one quick question, or maybe two quick questions.
So in the pairwise comparisons, the cultures that you did, the 100 pairwise ones, are you accounting for the influencer bacteria maybe themselves producing exometabolites that are harmful for the recipient bacteria?
Yeah, so we think that might, well, we think that a few things might be going on there. So we did not do exometabolite. We don't know exactly what they're producing. We do know that they're producing stuff. I didn't actually show that part of the data, but we do see production of other metabolite features by those bacteria. So that may be one of the ways that they are having potentially a positive impact through producing something new, through breaking down polymers. Or the other impact is that we know that this algae actually produces some inhibitory compounds, and so they may actually be breaking down a compound that inhibits another bacteria.